Hello, in this video we will discuss about the transfection. The process of transfection we will generally overview discuss. So the transfection is the process in which the introduction of new gene into the cell as a recombination to produce a product which that is not present before, and cell is known as transfecting cell which that is introduced a foreign gene. So that is, let's begin to understand the mechanism and we will learn about the transfection method. So first of all, we will need to understand here is the plasmid which that is extract from the bacteria will recombine by a foreign gene introduction. After the foreign gene introduction with the plasmid which that will be transmit into the cell, and the cell is recombinant cell or transfecting cell or transgenic cell will be occur, and the genetically modified, so that is why it is genetically modified transfecting cell is known as recombinant cell or transgenic cell. After this, it will produce a product either bind with the DNA in the genome or in the cytoplasm. The plasmid will remain in the cytoplasm to synthesis of protein after the transcription to translation of the RNA, messenger RNA. So let's begin to understand the type of transfection. It is, we can classify the three types. For example, the lipid base, calcium phosphate, electroporation. So first of all, the lipid base means the lipid will be a bind because it contains a positive charge, while the plasmid contain DNA is known as negative charge, and this interact to each other to form a endosome like formation is it will be enter into the cell via a endosome or the basically is the plasmid will release directly or indirectly. So calcium phosphate process mean the calcium will be bound because it is the cation, and it will move toward the anion. So in this way, here is the negative charge which that are present in the DNA will attract to the calcium to form a positive charge coat which that easily invade, and here is the electroporation mean the electricity will provide to create the pores, and pores will the cation will move out, positive charge will be move out, the proton will lead to attract the plasmid. So application of the transfection is, let's begin to understand the there are many application: recombination of genes, disease identification through reporter gene assay like the basically it is the luciferase reporter gene assay, and the biological product by recombination of bacteria, plasmid, and gene therapy by removing defective gene to introduce the gene of interest into the bone marrow or other cell, the stem cell, for proliferation to produce a normal cell rather than the defective cell containing defective genes. So that is the important things. The transfection is very important in the medical field. So let's begin to understand the transfection method. So this is the nucleus. Nucleus contains a DNA, genetic material, while here is the cytoplasm. So remember, this is the extracellular space, and the plasma membrane and nuclear membrane are present here. So the blue color lining show the nuclear membrane, while the plasma membrane are present in the outer. Here you can see brown color lining show. This is the bilayer of phospholipid, remember. So let's begin to understand the transfection method. Easy to understand. I will easyfied it or simplified. So the lipofectamine transfection 300, which that is used in this way the lipids introduced as a liposome, which that will be positive charge containing liposome bilayer of phospholipid like a plasma membrane, which that interact and to form a endosome formation. After this endosome formation, the plasmid will remain inside containing negative charge, while the positive charge is the outer coat, which that will attract toward the plasma membrane for the invasion, is known as endocytosis. So remember, this type of endocytosis is known as the phagocytosis because the not liquid particle but solid particle. So remember, so in this way, here is the endosome formation. This endosome formation, the proton will move inside to decrease the pH and increase the acidity. Will release the plasmid into the cytoplasm. Will go either into the nucleus or remain in the cytoplasm for synthesis of messenger RNA, as known as transcription, to produce the messenger RNA, and translation to produce protein. And that protein is target to the different function, for example the channel creation or the synthesis of insulin, like synthesis of hormone or synthesis of other protein, or synthesis of the other part of the cell for repairing. So to remember that organelle as well as other things. So here is a channel is able to movement active, passive, and transport is very important through channel. So anyhow, let's begin to understand the calcium phosphate transfection. This calcium phosphate transaction involve the calcium phosphate, which that contain calcium is the positive charge, which that attract to the negative charge as a plasmid, and in this way the plasma membrane contain negative charge will attract to form endosome same like structure, and the R plasmid will release. So remember, endosome if not formed, so the plasmid will release, and in this way the plasmid is the same function. But in the case of the fusion transfection involve the cationic polymers. This cationic polymers contain positive charge which attract the plasmid to form a conjugation like structure, which that will be move into the negative charge attraction in the plasma membrane to form of endosome formation, is known as the endocytosis. After the endocytosis, the endosome will be released, same mechanism, the pH will increase, lead to remove that plasmid. So anyhow, after the proton influx into the endosome, so here is you can see this is the positive charge coat creation. After this positive charge coat creation, it will move, and that is basically the same mechanism. So remember, the transfection is very important for a recombination of genes, disease identification through reporter gene assay, biological product of recombination of bacterial plasmid, gene therapy by removing defective gene to introduce gene of interest. So that is the a lot of application. Remember, which that is the transfection cell. Transfecting cell means the involving for transfection. So the transfection is the introduction of foreign gene. Thanks for watching.